This morning I will be giving you an overview of some of the work that my group and many other collaborators have been working on chikungunya almost 20 years ago. And you can see how the world is now embracing new outbreaks of chikungunya in several countries. So I'm Lisa, the current Executive Director of the Aedes Infectious Diseases Labs. And I'll just give you a little bit of a timeline. I see many young faces here, so maybe you may not be aware, but actually Singapore and many of some of the seniors here in the room have been in this field for quite a long time. I will just start after the year 2000 and maybe after the year 2000. I think before that my life was quite a blur like many other people. So you can see that in late 2003, that was a time when Singapore had the first SARS outbreak. Right? I think a couple of countries around Asia and also in Canada, I think there were reports. And it was actually one of the first episodes, a real life episode that actually made an impact to me, and also how it has shaped my views about using science to help public health and also clinical management. Now, thereafter, you can see that there were different episodes of vector-borne infections, bacterial infections, respiratory infections that continue to be one of the key disruptors in our daily life. And so, of course, right now, I think we are out of COVID. Right? Of course, I think many of us on and off will actually get some respiratory infections that happen to be COVID, but I think it's something that we deal with. But of course, as I mentioned in my first slide, of course, there are some new cases of chikungunya at the moment. So the Aedes infectious diseases was established right during the pandemic in late 2020 to early 2021. And the intention at that time was for Aedes to reconsolidate all the infectious diseases research groups and also to strengthen our part in Singapore's activities in dealing with the pandemic. And of course, the introductory video has sort of reminded us that we should not wait for another pandemic to do good science. I think this is when I think we are in a very good time at this point actually to reinitiate or initiate old or new collaborators in different parts of infectious diseases, and how can we better prepare ourselves for the future. It could be anything: viral, bacterial, fungi, or even parasite, or even host immunity. So you can see that at Aedes Infectious Diseases Labs focus on three research pillars: the vector-borne, respiratory, and antimicrobial resistance. And the various investigators will then bring together several core cross-cutting capabilities that will enable us to look at any infections. This is who we are now, and we are not at end state. I'm sure many of you recognize many of the faces there. So we have people who moved from Europe, UK, Australia, and also within Singapore, and we continue to look for new people to join us in our exciting journey. So you see they cover broad areas from viral, bacterial, fungi, and also Plasmodium parasite. So in terms of our focus, it's not exactly public health, but I will say that all the investigators have their own specific research interests. They have their pathogen of interest, or even certain host immunity pathways that they are looking at. But regardless, it's highly collaborative environment internally where we normally come together to answer a specific problem statement, and also we collaborate widely with the rest of the ecosystem in Singapore and also overseas. So in terms of the therapeutics, which you will be hearing, in different parts of the talk today, on host-directed therapies, on nucleic acid base possibly, and monoclonal antibodies, and of course the newer the newest kids on the block on AI drug discovery. So I think in terms of immunity, which is what we are talking about these 2 days, you'll be hearing a lot about antibodies, cytokines, T cells. I saw several talks on T cells, some of them on B cells. So what I would like to share today is just how some of all these research questions actually could come together actually to answer any challenges. And of course, we do have some infection models that I'm sure many people are somewhat familiar with. Okay, so if we were to focus in on the various pathogens, that's, as I mentioned, for vector-borne, we have viruses and also parasite. For AMR pathogens, the various investigators focus mainly on escape pathogens. And respiratory, I think, is something you will hear a lot this morning, coronaviruses and influenza. And of course, some of the other diseases that, again, will enable us to come together to, and if we tackle new challenges, right? So, of course, the focus on some of the vaccine capabilities are listed below, from epitopes, subunit, live attenuated, and also, of course, the RNA vaccines. All right, so I think in a post-COVID world, it is we all feel it. I think we are experiencing one this morning, the wet weather. So with climate change, inevitably, the mosquito-borne transmission will also increase. I think there have been reports of the various mosquito populations that are increasing their geographical habitat. Many countries that had never reported any cases of Aedes mosquito actually now report Aedes population, and that's a result. As a result, when you have Aedes population, there will increase the infections of dengue, right? Of course, that still remains the #1 vector-borne viral infection, and of course there are others like chikungunya. There's also the old nyong nyong in Africa. There's Mayaro in South Africa, and over in Australia, I think for many years they have been dealing with Ross River viruses. So I think this is where you can see that I think vector-borne diseases will not go away. It's just how we manage. Of course, there are other new modalities that control the vectors, but I think this is where what if it break through all those kind of measures and we still get bitten by mosquitoes, and what can we do? So a lot of work has been done actually on dengue, on T cells, and by Nina and groups at Duke NUS and many other places. But you can see that I think there's still a lot that we can come together to look at things differently. So just coming to chikungunya is something that I've been working on for a long time with many people in this room here. And so I will say that it's a very small virus. It's a small alpha virus of less than 10 kb, and again transmitted by none other than the Aedes mosquito. So I have to say that this was something that was first reported in the 50s, I think, for those who are not aware. And then there were sporadic cases over the 60s, the 70s, and then there were some that maybe just no one was looking at it. But well, in late 2005, 2006, there was a large resurgence of the chikungunya in the Indian Ocean islands, and that was because it sort of a jump into another Aedes vector, the Albopictus mosquito, and that was the one that actually caused the outbreaks that then got spread to India and then Southeast Asia. So it was at that time that myself and some other investigators started looking at CHIK with very limited tools and a lot of unknowns. So that was then, it was, I would say, a very fun and exciting time as well. When you are dealing with the unknown, it can be frightening, but it's also pretty exciting because anything you do, it's a piece of data. So I have to say that since then, there were also different episodes, right? There was, you can see the different clades, the different lineages. Of course, the one that spread to Singapore in 2008, it was the ECSA, what we call the East Central South African lineage. And of course, there was another resurgence of chikungunya quite unexpectedly in 2013 to 2014, and that is all the way here in South America. Right, and that one, strangely, is belonged to the Asian lineage. Okay, so how it got there, I think there were a lot of hypotheses. It was through the cruise ship. That was what the CDC hypothesized at that time. So you can see that with different things going on, then there was a new interest on comparing the different lineages, in terms of viral kinetics and disease phenotype and so on and so forth. So at this point, I would say maybe this was 2021, so I would say that it needs to be updated. If you're just looking at the alpha virus outbreaks around the world, you see it's quite busy, right? And you can also see that I was talking about climate change, where even in European countries, that you have cases of mosquitoes, and the moment you have mosquitoes, which means that there will be opportunities of mosquito-borne infections here. So it's not just a tropical problem, although we have a large population of people who tend to get infected from time to time. Okay, so as I was mentioning, this was since early this year there have been cases of chikungunya again, that reporting coming from the Indian Ocean islands, and then thereafter it started spreading to other parts. So I just want to bring to you this morning on all the different types of pieces of work that many groups have put together over the past 18 years for chikungunya since the beginning, how myself and the other groups have sought to answer the various important roles of interferon-related genes, and how the whole aspect of innate immunity will come together during early infection, and also the various stages of disease progression from early acute severe diseases, or what are the kind of markers that were very, very pronounced in a certain group of patients, and also for patients who then recover. In fact, everyone will recover, but there will always be a percentage of patients who have reported long chronic joint inflammation. So we have also identified certain factors that some of these patients would have. And also, while understanding the various mechanisms of protection, disease severity, it also enables us to establish relevant mouse models to study some of these mechanisms, and also develop new modalities of therapeutics, especially in the space of vaccines, right? So of course there is the epitope-based vaccine that was not only a very good detection marker, but it also it proved itself as a very interesting potential preclinical vaccine by just targeting this particular region of the E2 glycoprotein. And also the attenuated live virus approach, where I have to say that there was over 2 years ago, there was the new CHIKV vaccine by Valneva, which was also based on the similar approach, but of course the area is different. But of course, I think right now we heard that that particular vaccine was faced certain issues. But regardless, I have to say that the approach, I think it enables more potential to look for new vaccines. And also, while understanding the important roles of T cells and related pathways, it also enabled the team to identify some FDA-approved clinical drugs that could be repurposed to treat the joint inflammation during acute disease. So you'll see a little bit of this before I finish off with one of the latest study that allow us to look at a slightly different angle to control chikungunya. So at the beginning, I think this slide Antonio will remember, because I will always cite him, because he challenged me when I showed him this data many years ago, where I first identified the virus in the CD4 positive monocyte fraction. So of course he was saying maybe it's not specific, and all that. So I think with that, we then went on to do several series of heme inactivated infection assays to actually prove that no, what we have seen is actually specific, and also with with the same patient that we collected 66 days after the patient has fully recovered, you no longer detect the CHIKV antigen at that stage. So I think nonetheless, actually that enabled some early understanding of the target cells during the viremic phase during early infection. And of course, along the way, there were other reports from other groups from France, from Australia, from mouse models, from non-human primate models that also more or less provided complementary data sets. They have identified the virus in the macrophage populations, and also they found the virus through PCR in the joints. So by putting all the hypotheses together, this is where we showed that potentially the virus may hitch a ride with the monocytes, which then mature to macrophages, and they will get transported to the joints, where they cause the ongoing joint pains. I think that was the long question that we never completed, I have to say, because then CHIK went away, and we were looking at other problems. So along the line, same line I mentioned about innate immunity, right, with the patient cohorts at that time in Singapore, and also by comparative data sets from other countries, we also showed the important roles of toll-like receptor 3, as I mentioned, also some of the other interferon genes, such as viperin, and some of the other related groups reported from France, how they all come together to control the virus during early infection. And so I would say that it was also a very fun time those days, to identify, to confirm new pathways and all. All right. So, of course, with patient cohorts, we can only go that far. Then we had to establish a relevant mouse model. So, first reported by Andreas Serbier from Queensland. I think he has retired since, that he was the first one who showed that the virus could actually infect the C57 Black 6 animals, right? Because before this paper, all the other work was based on immune deficient animals, which is actually not the right model to study immunity. So, of course, we have since adapted this mouse model to use slightly younger animals, which recapitulated this outcome here, the peak of viremia, and also the peak of the joint swelling. So with that, it enabled a lot of very interesting collaborations and also understanding of the different mechanisms to really understand immune pathogenesis of CHIKV infection. So if we were to move on to the later part on T cells, so if you were to look into literature, pre-CHIK, a lot has been reported on Ross River virus, of course, a close related family of chikungunya, to be involved in, to actually use CD8 T cells. So actually we redid the same kind of experiments, in the CHIK model, but we actually could not find anything that significant when we did the infection in the CD8 T cell knockout animals. So on the contrary, it was the CD4 deficient animals that presented with a very significant outcome. So, that proved the importance of CD4 T cells instead. And subsequently, we then went on to confirm this outcome from with other different relevant models. And to make a long story short, we then explore, if we could use any FDA approved T cell therapies to control the joint inflammation. And actually, there are quite a few out there. Sometimes when you have too many choices, that's when it also can be complicated. So we went with the most obvious, and the most obvious normally are not the ones that are the right ones. So when we were about to give up, we tried, okay, let's do one more and see, if nothing, then we'll move on. But actually it was the third one, fingolimod, that proved to be the right one. So of course, at that time, it was very clear, with the treatment of fingolimod in the animals, all these animals no longer have have the joint inflammation. And you could also see from here, since we have already confirmed that typically it will be the CD4 T cells that get infiltrated to the inflamed joints. And when we look at the cells, when we did the immune phenotyping of the cells from the joints, you see that it has greatly reduced. So of course, this was again pre-COVID. That was pretty exciting. And now I'll move on to the last part, which is some newer work that was started pre-COVID. Then it got derailed for 4 years before the scientists could actually reinitiate this project again. And that was the angle of not just the host and the virus, because of what you have seen before, or all the work that we have done just between host and virus, and now we actually involve the mosquito, right? Because we need these three to come together. You know, that's how Laurent Rainier will normally explain when he talks about malaria and the Plasmodium. So we are using pretty much the same approach, but not that not so complicated, where the work was led by CY and also the collaborators at the National University of Singapore. So of course, CY is a senior scientist in the lab, and so her interest was really to look at any possible modulatory impact of mosquito salivary protein. So she was working with a couple of them, and there's one particular one that made the most advance in terms of progress, is this factor called salocinine. Okay, so this was the work that was initiated, I said, pretty long ago with through the work of Kini, who was actually who was retired already. But anyway, when they did the proteomics on the mosquito salivary glands, they actually found a particular protein that was actually very, very dominant, and also which is called salocinine. And when we then explored more to understand more of what salocinine is in terms of sequence, any motifs, so we found that it's homologous to the human tachykinins. And as a result, you know, for tachykinins, it's also well reported, you know, to bind to several human neurokinin receptors. So that was a very exciting find. So we wanted to then explore actually which NK receptors the salocinine will actually bind to. So of course there were 1, 2, and 3, and you based on some biochemical assays that were actually done by NUS by Vaishnavi. So actually we showed that it was the 1 and 2, NK1 and 2, that looked the most promising. So we continue on with those. And so you can see that when we treated salocinine on a primary human monocytes, you could see that it disrupted the performance. The signatures has changed. You can see that it actually reduces the activation of the human monocytes, especially the CD169, and also a lot of downregulation of the type 1 interferon signaling genes. And bear in mind, so these were the kind of genes that we have shown earlier by us and many other groups to be important upon CHIKV infection. Okay, so then we just move. I mean, of course, there's a big bound of biochemical cellular studies to prove the functions. I'll be happy to discuss with you on that. But nonetheless, then we went to the therapeutic potential, right, to actually just see how this performed in the animal model. So you can see here, by treatment or with salocinine, actually reduces the CHIKV joint inflammation, actually by suppressing the monocyte activation. You can see that it controls that, so the inflammatory signal, it's controlled. And also, if you look at the joints now, right, the joint inflammation is also significantly reduced. And also, but viremia in the blood, just like the earlier work on fingolimod, there's not much, there's no difference. So it doesn't control the viremia directly on site, as we could see from the PCR done on the joints. But of course, it's actually through the abrogation of the pathogenic CD4 T cells, as we have shown in the earlier model. So what she had done here was, of course, to collect the cells again, and then she did the ELISpot spot to look at the specific CD4 T cells, and you can see that the treatment actually reduced it significantly. And when she measured the pro-inflammatory and cytokines, again, you see that actually reduce the pro-inflammatory signatures somewhat. and that overall, it controls the disease outcome. Okay, so now I'll just finish off a little bit in the patients, right, because that was all done in the patient. So what is the relevance to the patients? Well, you can see from here that we then screen the CHIKV patients' antibodies against the salocinine, right, just to see if the patients had any antibodies against salocinine or not. And indeed, you can see from here, 97% of the CHIKV infected patients actually had detectable salocinine IgG. And what is interesting is that also the levels of IgG correlated with disease severity during the acute stage, actually, which fits into our hypothesis here. And I have to say that this study has been accepted recently, and we're very happy to continue the second part of some of this work. So I think, yes, just to close the story again, then when we re-look at all the systemic inflammatory markers against the levels of cytokinin, you can see that there's a positive correlation. So I think just to finish off, as I mentioned at the start, we are experiencing new waves of CHIKV outbreaks. It may not be as severe as we think they might be, but in case they could be, I think we need to be prepared. So I have to say that with all that has been done and also lessons learned from COVID, it actually enabled the teams to come together faster. We know of one another's expertise and how can we actually do things together to answer and improve some of the current limitations, such as the detection, the differentiation between, to distinguish between dengue and chik. I think because it's known, in fact, this problem was brought up during the Zika outbreaks where there's a lot of cross-reactivity when we're doing serology assays. I think this is an area that we can still sharpen. And also, has it changed in terms of mosquito species? So right now, of course, both species will transmit both the Aedes aegypti and the albopictus. And what else? And, with the, are we able to identify the populations at risk? I think these were lessons learned during the COVID. I think that could be re-implemented in another disease setting between the elderly and also the young. And so with that, I would like to thank you for your attention. This is the team, and also for the various collaborators all over from different parts of the world. And so thank you for your attention.
Thanks, Lisa. Great talk. I'm just interested, with the anti-salicyline antibodies, do you think that they're increasing inflammation by actually cross-neutralizing endogenous kinins that are playing some sort of modulatory role rather than necessarily blocking the...
Very interesting question. I think that's something that we are currently working on to understand that part. Our belief that it is also the timing, whether, we have to look at the acute where, probably increase, but actually what happens, it could be what you just mentioned. We don't know at this stage, but it's something we are looking at.
Great.
Thank you.
Yeah, Lisa, very nice talk.
Yes, thank you, George.
I wonder, for your salocinine, are there 97% of the chikungunya infected patients, they would have IgG. Any evidence, for all the chikungunya recover, they, because of salocinine, they would, if they have the following bite with a mosquito, any evidence to see, their inflammation would be reduced whatsoever?
Of course, we can only do by association based on the levels of salocinine and also the CHIKV, IgG, and also the phenotype. At this point, maybe we don't have enough numbers. It's not so clear as what you just mentioned. It's not.
Yeah, I think if it does, that is will be a very good immunology candidate. So universal vaccine for any mosquito-borne disease.
Even dengue. I think that was the dream we had, whether the airy-fairy dream. But I don't think it's so straightforward. That is not the only magic bullet. It could be one of the many things that may impact, and also it's also the timing. Yeah. I think
any other proteins in addition to this?
Yeah, there are some others, but the behavior is also very different, not quite the same.
Yeah. Thank you. One question also for the salocinine. So it's in the saliva of the mosquito, but only if the mosquito is infected with chikungunya, or every mosquito bite with a totally healthy mosquito will give us antibodies?
Yes, I would think that any mosquito, which is why I think in our population here, we all will have levels of salocinine. Is just high or low, and does the level will impact the infection? That we don't know. Very good question, yes.